I'm Zhongqi Lu. I'm a professor at Department of Cell Biology and Molecular Genetics at the University of Maryland. So my lab is interested in understanding the basic molecular mechanisms that control flower and fruit development. The fertilization of the flower is really important for the seed to develop, and the seed development is important for the fruit to develop. So we are very interested in understanding how fertilization triggers the seed to produce a signal and how that signal is transported to the fruit tissue and how the fruit tissue sends that signal and respond to that signal. By understanding these mechanisms, we hope to translate the knowledge into solutions to agriculture.
My name is Rachel Shahan. I'm a graduate student in Zhongqi Lu's lab. This is my fourth year of my PhD, and I work on strawberry, flower, and fruit development. So in our lab, we're particularly interested in a diploid strawberry called Fragaria vesca. Fragaria vesca is a diploid variety, so that means it has 2 copies of every chromosome versus the strawberry you would buy in the grocery store, which is Fragaria ananassa, and that is an octoploid and has 8 copies of every chromosome. So it's much more difficult to do any sort of genetics work on an octoploid because you would have to, for example, knock out a gene 8 different times versus only 2 times in the diploid. So in strawberry, it's a pretty exciting little fruit. It's really fun to work on. One of the most interesting things about it is the true fruit is actually not the fleshy part that you're eating. The true fruit is called an achene. So if you've eaten a strawberry and you've seen these little kind of dots on the surface, they're like little seeds, that is actually the true fruit. So in each true fruit contains 1 seed inside. The fleshy part of the strawberry is actually an enlarged receptacle. So in strawberry it is actually particularly interesting because the seeds are on the outside, so it's an inside-out fruit. So this is interesting because you can study how the seeds affect the development of the fleshy fruit really easy because you can see it happening right before your eyes. So here, for example, we see only 1 fertilized achene. So in this case, only the receptacle tissue directly subtending that achene is developing.
In 1950s, a professor at Harvard called Nish did some experiment where he plucked all the seeds away except 1 or 2 or 3 seeds left, and observed that only small patches of fleshy fruit grow underneath that leftover seeds. And so he reasoned that the seed must be secreting some growth stimulating molecules that enable the underlying tissue to grow into fleshy fruit. So later on, he was able to pluck all the seeds away, but then spray a plant hormone called auxin onto this fruit. And now he found out the auxin can substitute the seed. So he reasoned it's likely the seed is responsible for secreting the plant hormone auxin to stimulate fruit development. Nish's experiment indicate a fertilization-induced signal produced by the seed is important for fruit development. If we can understand this process, especially the underlying molecular process, then we can maybe hijack the signal and use the signal to tell fruit to develop even in the absence of fertilization. So in order to understand how the diploid strawberry flowers and fruit develop, we first determine the developmental progression of flower and fruit development. By understanding the morphology, the development of this developmental process, then we can better look at the molecules, the molecular events underlying the developmental progression and allow us to eventually identify the genes that control these processes. After the first experiment to show the wild strawberry exhibit the same phenomenon as what Nish observed for garden strawberry, then we moved on to undertake a serious experiment to get at the question about what are the molecular events underlying fertilization-induced fruit initiation. So we first actually did a detailed descriptive work on how the flower developed, how the fruit developed, so that gave us landmark events. We have at least 25 different fruit tissues and about 13 different floral tissues that we have made a library and sequenced them. And then subsequently, we analyzed the expression of the whole genome of the expressed genes during flower and fruit development. And we were mostly interested in looking at hormone genes like auxin biosynthesis genes, auxin receptors, and auxin signaling pathway genes to see how their expression are regulated during flower and fruit development.
So the idea behind the transcriptome is, instead of looking at gene by gene how genes are expressed and where they're expressed, we can get a global view and look at where and when all the genes are expressed. So all of our data is actually available online in the Strawberry Genomic Resource database. So if you go and click through and just look at what our data looks like, you'll see the annotation of a gene, and then all of these little reads below are RNA-seq reads.
So based on this initial analysis of transcriptome data, especially the biosynthesis and signaling molecules in hormone auxin and GA, we proposed a model about how fluid initiation is achieved, mainly the fertilization of the central cell, that's the precursor to the endosperm. That secondary fertilization event stimulated auxin production, and very likely the auxin production also stimulated GA production. And both of these hormones are being transported to the underlying fluid tissue and perceived by receptors in the underlying fluid tissue to initiate the fluid development.